Welcome to this explainer. Today, we're tracing the incredible evolutionary journey of our genetic material, going all the way back from its ancient origins right up to the absolutely mind-blowing microscopic machinery that copies it today. If you've ever wondered how the complex blueprint that makes you, well, you, is preserved, protected, and perfectly duplicated millions of times over, you are definitely in the right place. Let's get into it.
Okay, so here's our roadmap for this explainer. We're going to hit 4 main stops. First, the ancient RNA world. Second, semi-conservative DNA replication. Third, the heavy nitrogen experiment. And finally, we'll wrap up with the cellular copying machinery.
Starting with Chapter 1, the ancient RNA world. We are going way, way back, billions of years, to the very first origins of our genetic code.
Now, to really understand DNA, we actually have to look at its predecessor first. Science tells us that RNA was the absolute first genetic material. You can kind of think of early RNA as a super ambitious startup founder, or maybe a molecular Swiss Army knife. In the ancient world, it literally did everything. It wasn't just sitting around storing genetic information, it was actually out there driving essential life processes, like metabolism, translation, and splicing. It was the genetic code and the active catalyst for biochemical reactions all rolled into one.
But here's the catch, and the contrast between these two molecules is just fascinating. Because RNA was acting as a catalyst, it was highly reactive. And well, in chemistry, being reactive usually means you're chemically and structurally unstable. It was just doing way too many jobs at once. So molecular evolution stepped in and ultimately created DNA. DNA evolved from RNA with these chemical modifications that made it far, far more stable. Think of DNA as a secure, stable hard drive of the cell. Because it's double-stranded with those complementary strands, it inherently resists changes, and it even allows for some pretty sophisticated repair processes to keep the data safe.
Moving from the highly reactive RNA to this ultra-stable DNA hard drive begs a pretty monumental question, one that definitely puzzled early scientists. If the DNA double helix is locked up so tight to prevent mutations, how on earth does it manage to perfectly replicate its code every single time a cell divides?
That brings us to Section 2, semi-conservative DNA replication. Let's look at how researchers first wrapped their heads around duplicating this complex molecule. Back in 1953, when James Watson and Francis Crick proposed that famous double helix structure of DNA, they confidently dropped this legendary and honestly famously understated line. They noted that the specific pairing they postulated immediately suggested a copying mechanism. Basically, they realized that the rule of complementary base pairing, where adenine always pairs with thymine and guanine always pairs with cytosine, naturally provides a direct template for copying. If you know one side of the DNA ladder, you automatically know the other. It's built right in.
And their model brilliantly illustrated this semi-conservative approach. The idea was that the two parental strands separate, and each one acts as a template for synthesizing a brand new complementary strand. It's exactly like duplicating a rare, ancient recipe book. Imagine carefully taking the pages apart, keeping one original handwritten page on the left, and then binding a brand new, perfectly copied page right next to it on the right. So after replication, every single new DNA molecule contains exactly one old parental strand and one newly synthesized strand.
Section 3, the heavy nitrogen experiment. Now, an elegant theory is great, but it always needs concrete proof. And that leads us to a literal forensic-level investigation in 1958 by Matthew Meselson and Franklin Stahl. To prove this semi-conservative model, Meselson and Stahl basically played baker. They baked cellular bread. First, they grew E. coli bacteria in a medium containing a heavy isotope of nitrogen, 15N. Think of this as feeding the bacteria heavy flour. So all of their DNA was built using these heavy ingredients. Then, they abruptly transfer those cells into a normal, light 14N medium. As the cells multiplied, they extracted the DNA and spun it in a cesium chloride density gradient. Now, this is a technique that separates molecules purely based on how heavy they are.
And the result? Well, look what happened after exactly one generation of cell division. The extracted DNA didn't settle at the heavy band at the bottom, and it didn't float up to the light band at the top either. It settled right smack in the middle. It had a perfect hybrid density. This proved beyond a shadow of a doubt that exactly half the DNA was from the old heavy ingredients and half was from the brand new light ingredients. Boom! The semi-conservative theory was absolutely correct.
Section 4, the cellular copying machinery. So, having proven that the DNA safe perfectly splits and copies itself, we've got to zoom in on the actual microscopic factory workers that are doing all the heavy lifting.
Prepare to be amazed by this number. In simple E. coli bacteria, which have 4.6 million base pairs, the entire process of replication finishes in just 18 minutes. That means the cellular machinery is operating at a mind-boggling average speed of 2,000 base pairs per second. 2,000! And it does this practically without making mistakes, because any error would result in a permanent mutation. The real MVP behind this breathtaking speed is an enzyme called DNA-dependent DNA polymerase. This enzyme is basically a high-speed, highly accurate typist, just sipping down the genetic code. But as you can imagine, typing that fast requires an immense amount of energy. So the raw materials it uses, deoxyribonucleoside triphosphates, actually pull double duty. They act as the letters being typed into the code, sure, but because they have these high-energy phosphates attached, they also provide the actual power for the reaction. They act just like ATP batteries.
Now, our master typist does have some very specific rules to follow. It doesn't just randomly start copying anywhere it feels like. It always begins at a very definite genetic coordinate called the origin of replication. Also, unzipping the entire massive DNA molecule all at once would just require way too much energy, so instead, the DNA only separates in a tiny, localized opening. We call this little Y-shaped opening the replication fork.
This is where the mechanics get incredibly fascinating and a little chaotic. Our polymerase typist can only move in one single direction, 5' to 3'. Imagine paving a two-lane road, but the paving machine can literally only drive north. On the lane heading north, the paving is perfectly smooth and continuous, but on the lane heading south, the machine has to keep backing up and paving in these messy, disjointed patches. In our DNA factory, the continuously paved side is one template and the disjointed patch side is the other. To fix that mess, a completely different worker enzyme named DNA ligase has to come in right behind and seamlessly glue all those discontinuous fragments together into one solid strand.
So, we've seen how early life transitioned from a chaotic RNA world to the incredibly stable double-stranded DNA molecule. We've explored how it elegantly unzips to preserve its own history, and we've peered inside the microscopic, high-speed factory that paves new genetic roads with just astounding accuracy. But this leaves us with a pretty profound and fascinating question to ponder as a gateway for your next learning adventure. If DNA is just a locked safe full of chemical letters, how exactly does this internal code get read, translated, and expressed into the physical, living, breathing traits of every creature on Earth? Keep that in the back of your mind as you continue exploring the molecular basis of inheritance. Thanks so much for joining me on this explainer, and keep learning.